Mike mentioned, I've been working on habitat suitability modelling recently in the last few years. Especially at the moment, I'm working on a project called RESO, which stands for Restoring Seagrass for Ocean Wealth. And it's part of an SMMR project, which is the Sustainable Management of UK's Marine Resources. And the point of the funding for this project is really based on the fact that the UK government has got commitments to meet net zero climate change targets. And as part of this, nature-based solutions such as restoration are coming to the forefront of research. And so the main aim of this project is to come up with a coastal ecological enhancement decision support tool for future restorers or practitioners that want to restore seagrass meadows. And some of my work will be feeding into this tool in the future. So for those of you that don't know what habitat suitability modelling is, it is a method of relating biodiversity or species and observations at where they are found at the moment to prevailing environmental conditions. So looking at what the environmental conditions are of where species actually are in occurrence. We then use algorithms, mathematical algorithms, in order to explain those links between biodiversity and the relationships between where the organism is found and the environmental variables, which explain where it is. And this is all based on niche theory. It enables us to predict where species are present at the moment, where they could be found, or where they might be found in the future in relation to future climate change. But it's also used as a way of finding where suitable areas could be for restoring of certain habitats or for species management and conservation. So there's lots of different methods that are used for habitat suitability modelling. It's also sometimes called species distribution modelling, and it will use lots of different types of algorithms can be used. They can be based on profile, regression, they can be non-parametric machine learning methods. There's loads and loads of different types out there. It depends on what kind of data you've got as to which algorithms you might want to choose. For example, if you've only got presence data, so that means you've only got positions or observations of where that organism or species is found, but you don't have exactly explicit absence data showing you where it's definitely not found, because that can be quite difficult to collect in the field. Then you might want to use just a presence only algorithm, such as ecological niche factor analysis, maximum entropy, and so on. If you've got presence and absence data, you can use more regression-based models such as GLM and GAM. But you've also got the ability to use things like pseudo absences, which enable you to use a different range of algorithms. This basically creates pseudo absences. So you put background points based on your geographical locations, or it can be just randomly pinpointed in the spatial area that you're looking at for your habitat suitability model. There's also the option to use an ensemble of methods, and then this will take a weighted average of all those different model outputs, and it takes out some of the subjectivity of choosing one method over another. So that's quite a popular method to use. So a little bit about why we might want to restore seagrass meadows. So first of all, what are seagrasses? Some of you might not know what they are, but I think a lot of you do know very intimately what they are. So seagrasses are marine angiosperms, which means that they're flowering plants. They can form vast meadows, and they have really extensive root and rhizome systems. And because of that root and rhizome system, it means that they can actually store a lot of carbohydrates, and therefore quite important as carbon stores. They're ecosystem engineers. So the leaves of seagrass meadows within the water column, they can actually slow down and baffle water movement. So this will slow the water movement down and will allow the settlement and sort of filtering of particles out of the water, which actually makes the water clearer, which is really important if you're a marine plant. because you need light in order to photosynthesize. And so they're really important habitat forming species. And I'm concentrating on the species of Zostera marina, which is the most extensive meadow forming species we get around the UK. And this is Zostera marina, picture of it there, or also known as eelgrass. So why do we want to restore it? We've had huge losses over the years, up to about 92% around the UK, they think, as seagrass meadows have been lost. So in one way, it's just good to get those seagrass meadows back, but also they're really important carbon stores. As I mentioned, the extensive root and rhizome system means that because they're photosynthesizers, they will take carbon dioxide out of the water column. And then through growth and spread, rhizomal spread, they will actually store that carbon. And when they die off as well, those dead leaves will actually get held up into the sediments and stored in the sediments over time. They also are a really important habitat for many fish species, including commercial fish species, and they support a range of biodiversity, so really important in supporting biodiversity of our coastal waters. And as I mentioned, that root and rhizome system provides stabilization of substrates as well. So it will stabilize sediments, enabling us to sort of, it actually increases, or it decreases, I should say, coastal erosion or sediment erosion as well by stabilizing those sediments. So for habitat suitability modelling, you need to know what sort of environmental conditions or environmental data you want to put in your models. So these are some of the main environmental conditions that Zostera marina or seagrass would require to live. So they need a minimum amount of light. They need nutrients, like all plants do, to grow, shelter, salinity, because they're a truly marine system, marine species, I should say. Although they do tolerate a range of salinities, and quite often they'll grow in estuaries, for example. Temperature range will affect their respiration, for example, so they can live within a range of from 0 to 30 degrees Celsius. They need soft, sandy or muddy substrates in order to be able to grow their roots and rhizomes within those, and they need oxic conditions for their roots and rhizomes. And then also CO2, as a plant, they require CO2 to grow, but that's usually not a limiting factor. So when I started doing habitat suitability modelling, we wanted to look at what the best methods were to use. And as I mentioned, there's loads and loads of different methods that could be used for this modelling. And so we did a review of different modelling methods for marine macrophytes. We found there weren't that many studies that were done on just seagrasses, so we expanded it to marine macrophytes, including algae and wetlands, and then looked at those studies to see what variables they were looking at for their modelling and what model methods they were using. And we published this a couple of years ago, and we found that the most popular method was using actually an ensemble of different methods, so using lots of different methods, taking an average, followed by maximum entropy, another method that was really popular in the studies. And then this graph just shows some of the most common environmental variables used in habitat suitability modelling for macrophytes. The orange shows the macrophytes overall, and then the blue, the blue studies, the orange are just the seagrass studies. But we also found that this could be just that these are the variables that are most widely available. So they're not necessarily the most important. Quite often they are, but they might be just the most widely available variables, which is why they've been chosen. So based on our review, we then went on to test some habitat suitability modelling. We've done some projects looking at localised areas around Wales, which I'll come to in a moment. But for the project I'm working on at the moment, we are looking at different case study sites around the UK, but also the UK and Ireland as a whole. So in order to carry it out, we need to get really good presence data of seagrasses at the moment, and or from past surveys as well. But knowing sort of where the seagrass is at the moment in relation to environmental variables that we can get. That's really important in order for us to be able to predict where they might be able to grow in the future. Some of the variables that we were able to find and to use included light, or PAR, light availability at the seabed, bathymetry, temperature, salinity, wave energy at the seabed, and energy at the seabed due to currents, and also slope that you can derive from the bathymetry data that you get. So these are the variables that were consistent for the whole of the coast of the UK, which is why these ones were chosen overall. There are other variables that we could have included, but they weren't consistently covering all of the case study sites, so they weren't included at this point. The method we chose is an ensemble method. So we chose these different variables, these different algorithms, sorry. So we wanted to choose a mixture of machine learning methods and regression-based methods, and some of the most commonly used and tested, tried and tested from the literature. We did 10 repeats of each method as well. And then the ensemble will take an average of all of those different algorithm outputs, and for each of those 10 repetitions. So 10 repeats of each of those different model methods. You're able to look at the separate model outputs as well if you want to dive a bit further into it and see which model methods are performing better than others, which is what I'll show you in a moment. And then we created a model for the whole of the UK, as well as separate models for the different case study sites to do a bit of a comparison for those. So model performance for our case study sites, they performed well as a whole for the UK and the different smaller sites. 2 sites we found that didn't perform so well, or some of the algorithms wouldn't converge for a few different reasons. So for Craignish and Portincline, 2 areas, one is a small area in Scotland and one's a small area on the north coast of Wales. I think what we found was that the areas were so small, a lot of the presences were very clustered, and so there weren't enough presence points, and so not enough data and not enough variation in the environmental factors in order for us to make really good predictions of where else it could grow within that small area. So this graph is showing some of the statistics that come out of the habitat suitability model that enables you to look at model performance. So you've got area under curve, the correlation coefficient, and true summary statistic, which are the main ones that people will look at to look at model performance. And the higher the score, the closer to 1 of each of these, the higher or the better it's performing, that it's converging. So overall, most models performed well, but we did find that random forest performed the best, and GLM was slightly the worst, but overall they were all pretty good, I can say. When you look at variable importance, so this is looking at the different environmental variables we used, it changes a lot between the different sites that we looked at, and I'll show you a map of these different sites in a moment, which might explain some of these things. So for example, PAR, which is the photosynthetic active radiation, or the light at the seabed variable I mentioned, this seemed to be the most important variable for the UK as a whole, and also for Orkney. And it sort of makes sense because they're plants, they need light in order to photosynthesize and to grow and exist. Bathymetry was then the most important for Milford Haven, Studland and Craignish. Temperature for the Isles of Scilly, which is interesting. Salinity was most important for the Solent and Portincline, but specifically salinity in August. So some of the data variables that I used were actually temporal data as well. So some of the results, habitat suitability model will spit out a really nice heat map. So basically you're looking at a spatial area, which is where you want to look at where your presence, where your data is, where you've got actual presences of a species, but you want to look and predict where in a wider area that species would potentially be able to be in existence. And the whole reason of doing this is because it's very difficult to go and survey massive areas of coastline or whole countries, for example, for looking for a species. So you want to do this in order, or this is used in order to help you maybe refine where you might want to carry out future surveys, for example. So these are the case study sites that we looked at, and these are heat maps. So the more red the colour, the more suitable it is for seagrass growth, for Zostera marina to be able to exist. So it gives you a probability from 0 to 1. And you can create these heat maps, but also you can create a threshold as well if you want to, based on other statistics that you might want to investigate further in order to create that threshold, or you can just go, well, I want to have a look at what's more than 50% likely for seagrass to be able to survive in this area. So a lot of people will look at only those areas that are over 0.5 probability of suitability or even higher. So just going back there a moment, I mentioned 2 of the sites didn't do very well, and that was Loch Craig Niche and also Portstentline. We think it's because some of the, for example, Loch Craig Niche, some of the environmental variables didn't actually cover all of the site that we're interested in looking at. So, for example, the light data that we had didn't cover where the seagrass already was, and so we weren't getting good enough results when we included light into that model. So we had to take light out in order for it to work. And then Portstentline, it was such a small area, I think, that spatially, where it's showing that seagrass looks like it is likely to be, or its highest probability is exactly where the seagrass meadow is already. So it's not really giving us any new ideas or predicting new areas of where you might want to plant seagrass within that bay. But I think that's probably due to the resolution of the environmental variables that we had, and also the clustering of the presence data within that area. So we didn't have much variation between where it was found and the environmental variables and so on. So a previous study that we did, we wanted to drill down and look at these smaller areas, some smaller sites and smaller areas, and focus habitat suitability modelling to be able to sort of look at where within this small area would you want to plant, say, one side of a bay compared to another. And so we did collaborate with our coastal engineering colleagues, and they created some Delft 3D wave models for us to get some really fine scale hydrodynamic data or wave height data, which we inputted into some habitat suitability models for these different areas, as you can see in this figure down here. And we found that the fine scale data did change the variable importance overall for the habitat suitability models. We found that when the lower resolution wave data was used, then we had bathymetry and slope were the most important variables predicting Zostera marina. But when you had the high resolution wave data used, then wave energy became the most important variable for predicting where seagrass was likely to exist or suitable for seagrass to exist. We've also done this in another site. We've looked at the Isles of Scilly, and here we've worked with the Coastal and Marine Applied Research team down there, who created lots of hydrodynamic models, model data, looking at loads of different types of hydrodynamic factors that we could then put into habitat suitability models. And we did another similar comparison. If you look at A here, the figure A, that's showing an outline of sort of the Isles of Scilly area. And this is a habitat suitability model output from just using open source, relatively low resolution environmental data, compared to when we use some of the hydrological parameters that they created for us. And it really refined where it looked like it's suitable for seagrass, which is much more, looks like a much better prediction based on like the knowledge of where seagrass is actually found in the Isles of Scilly. So it makes a lot more sense, the figure in B. And so next steps, we want to use some of this, and it's already being used with Project Seagrass. Have been using some of my habitat suitability models to look at or to help inform where they might want to do some of this restoration. Quite often the models that I've created, because it's looking at subtidal Zostera marina, it's not always been the most helpful for them at this point. I'm hoping in the future that my models will be better improved and help inform practitioners like Project Seagrass to help them carry out seagrass restoration in the future. I also want to look at, well, what I will be looking at as part of my role, looking at climate change scenarios and using data from climate change scenarios, how that will change the outputs of habitat suitability models, and the potential for looking at using these methods for things like biodiversity. Can we use habitat suitability modelling or species distribution modelling for trying to sort of assume or get values for where biodiversity is likely to be highest, dependent on other factors or other environmental variables as well, which I think will be another important factor going into our nature-based solutions that we are tasked with improving and working on. So thank you for listening.
Mind if we save questions for the end?
Yes.
Yeah. So there we go. We'll just pass straight on with the next talk, and hopefully we can save up questions for Chiara after that, if that's okay. Right, I'll introduce myself, if that's not too rude, very quickly. I'm Mike Fowler. I'm an ecological modeller based at Swansea University. Although I kind of principally work with mathematical models, I've always got an interest in talking to people who work with real world things, and that's why I think a program like this is ideal, you know, allowing us to step out of our own little silos and forcing us to talk to each other, empirical and more mathematical ecologists anyway. So I'm really pleased to be part of this series of seminars. I'm going to talk today more about seagrass and some mathematical modelling we've done, largely by Dr. Jane Albright, who's since moved on to another position, but along with Jim Bull as well. We've, I'll accept any responsibility for any confusion or errors in the presentation I'm about to give. Jane did a phenomenal job putting all this together. And we've already covered some distinctions in this series of talks between restoration and rewilding and maybe forewilding. And I was trying to come up with an appropriate title, and maybe planting is as close I can get without offending somebody or many people. But let's see how we get on with that. So the first point I want to make is related to some very old work now of mine and some colleagues, where we looked at simple kind of competitive communities and the effects of removing species from those communities, which can trigger secondary extinction cascades. And then Per Lundberg and colleagues looked at trying to reintroduce the species that was removed previously. And they found that sometimes you get this community closure. You just can't. Some of the time you can reintroduce it, but because the community has changed since it was removed initially, sometimes it's just not possible to put it in. And I explored this a little bit further with Jan Lindström as part of my PhD, and we discovered that, in fact, sometimes it can be worse than that. It's not just that the removed species can't be reintroduced successfully. It's that sometimes that attempt to reintroduce it can lead to further extinction cascades, which is what I'm showing in the bottom right panel here. So there's, you know, a reasonably high probability, even in relatively small communities, 10 species communities, that attempting to reintroduce species can trigger further extinctions. So we need to be aware of this in a lot of these discussions. But it's also a reason why the seagrass question is quite good, because it maybe seems a little bit safer. If we're reintroducing seagrass, maybe we're starting from a clean slate and we don't need to worry about kicking further species out of the system. Maybe it's not as easy as that. That might be a typical modeler's oversimplification. So in this project, some of the key questions we wanted to examine were things related to what people actually go out and measure in the field. Simple things to measure, like the number of species that are there, or the Shannon diversity of the community that's there, or different sort of measures of diversity or complexity, as James Bullock spoke about in one of the previous talks. And so we looked at a range of different global species pools. So a kind of soup of species that are in the sea that might arrive or will arrive eventually at the newly planted seagrass meadow that we're focusing on. We also wanted to understand how quickly these communities will recover following the introduction of the foundation species, the seagrass species, so that we'd get some kind of idea about recommendations we could make. If you've got a limited amount of money to spend on sampling and following up your rewilding experiment or your restoration experiment, when's the best time to go out and collect data to find out if it's actually worked? And this relates to ideas that Sergey and others have been talking about and promoting recently around transients in ecological systems. And finally, something that I've not had time to dig into yet, but which I think is an interesting question, is if we dig down into different trophic levels, do we see the same answer that we'd see if we looked at the community as a whole, or do we see different patterns across different trophic levels? So, as I said, that's an interesting question, but we've not quite got there yet. So the simple mathematical methods that I've used are a kind of logistic growth, multi-species logistic growth. This is sometimes a kind of extended lot cut Volterra type model that we refer to. In this case, I used we used four trophic levels, and we based the ideas of who was in each trophic level on. Yeah, sorry. Here we go. Let's see. There you go. Yeah. We've got, it's based on relatively local literature, although none of the literature all comes from the same place. It's all spatially distributed. So there's maybe some arguments or some discussion about whether that's the right way to do it, but it's a starting point. It's a point that's come up over and over in these talks. We don't have enough data to account for all these uncertainties. But that's something that these kind of models can help us address a little bit. So we'd have apex predators, things like crabs or sea spiders or sharks. They have a very high mortality rate, or the highest of the mortality rates, let's say, so a very low RI value, intrinsic growth rate or mortality rate. We'd have mesopredators, which have slightly lower mortality rates, things like starfish and worms and anemones and some other fish species, teleost fish. And then the herbivore or grazer trophic level have lower still mortality rates. No, is that a higher? No, it's a lower mortality rate. And then finally, we've got our basal species, which is Zostra marina in this case. And in fact, this blob, this green blob down at the bottom of the food web here, really represents multiple species. It's a combination of green stuff that everybody else is feeding on, or the herbivores at least are feeding on. And again, we can base some ideas about carrying capacities for this on the literature and come up with some hopefully agreeable numbers to plug into the model. Do I want to say anything else about this? Let's see. Oh yeah, so there was some randomization. We replicated these communities for each different community size. We would replicate with different strengths of interactions across the trophic levels. And we'd also, in that replication, have different connectances or different trophic structures across the trophic levels. Yeah, hopefully everybody is managing to consume something on a trophic level directly below or a couple of trophic levels below or more if you're an omnivore. And the interaction strengths or the energy flows that we've got are based again on some limiting factors or limiting numbers that we can pull from the literature. There's some uncertainty around those, but we can say that the effect of a species I or a prey species or a resource species on species J, which is a consumer species, is drawn from some random numbers from a uniform distribution between two limits, positive numbers here. And then well, maybe that should be the other way around. The conversion efficiency is this parameter C here, is just the opposite of the other trophic direction. And that tells us how much benefit a consumer gets from its prey species. Okay, we've also got other initial conditions to put into the models based on some biomass density, biomass per unit of area. For the seagrass and the consumers, the seagrass is initiated at a relatively high initial density compared to the consumers. There's, as I said, some background soup of each of these other species that we've got in the food web. And it might be larvae that float past and settle in the seagrass eventually, or it might be an adult that swims through and starts nibbling on things. What else do I want to say? I think I mentioned about the baseline mortality levels. We used a minimum detectability in our model. Again, thinking about what people might actually be able to see if they're out diving at a site. If the local abundance of something is really small, then we're assuming that they won't see it. But if it goes above some threshold dynamic, then we will start to count that that's there. If it's below the threshold, then these species can still influence the dynamics. So we're not excluding them completely from the picture, but we're just saying it's going to be really difficult to count them and include them in the statistics that we're collecting from this sort of system. We used a simple simulation approach for the first 100 years to find out what was happening in the relatively early stages, though I'm not sure of any data collection that's gone over 100 years for a rewilding experiment. I think Lisbeth mentioned, or maybe one of the other talks, mentioned, 40 and 50 year series that people have, which is quite impressive, I think. But we're maybe not quite at 100 years of data yet. And these are the sort of simulation results we can get. I've got a log x-axis here, so time speeds up as we move along the x-axis. We start to see low densities increasing over time, or this is actually the number of consumer species that we find in these systems, and we see it leveling off after 10 to 20 years. And if we start with a global species pool of 8 species, we maybe end up with 3 or 4 species typically after 20 years, and that stays fairly constant until about 100 years. If your global species pool is larger, then you end up with more species by the end of it, but they all show a fairly similar sort of pattern where after 10 to 20 years, we're starting to asymptote, we're starting to level off in the accumulation of species in the system. And we started at a small 8 species food web and went up to a 57 species food web in this. And they're all there in the background. Some of them might be below the detectability limit, but then if they are below the detectability limit after 100 years, then they're not having a great impact on the dynamics of the system anyway. And we also looked at the much longer term dynamics of the system, and we rested on some really nice work by Wolfgang Janssen and Richard Law and collaborators looking at permanence theory. So this is bringing in another mathematical approach to answering some of these questions. I'm not going to go into this at all in any depth here, but it just allows us to see, given the background species pool that we've got, what are the permanent and uninvadable sets of species that we will end up with at the end of this. And there might be more than one, depending on the order that species arrive or different sorts of effects like that. But it's another nice mathematical framework that we can use to give us more certainty about what the final community might look like, without having to simulate it through all sorts of different initial conditions and taking lots of time like that. Yep. These are great papers to go back and look at if you want. Hopefully you can pick up the references in the recording if you need to. So yeah, mentioned already, we can count things like the number of consumer species present. We almost never got the full complement of all possible species after 100 time steps. But as we increase the global species pool, we also find an increase in the final number of species that we ended up with. There's a reduction in the proportion or the percentage of the final or the total species pool that was there as we increase the size of that species pool. So that's one interesting result. In small pools, we get almost a third of all species there. In large pools, we're tending down to 14–15%. Fewer of the total species pool is there after 100 years. We could also look at other measures of diversity or complexity, like the Shannon index or Simpson's index, and these all show very similar patterns to just the simple number of species that were present or detectable. The larger your species pool, the higher the diversity of species that you get at the end of the 100-year simulation. We also noticed some interesting transient patterns, that we would get some species appearing, you know, in the first 10 or 20 years, or maybe even a bit longer, that were never going to be in the endpoint community. So they would be there for some short period and then eventually disappear from the system. And then we'd end up close to our maximum final community size after 100 years or so. We asked how many of the final communities were unique in this persistent or uninvadable endpoint framework. And sometimes you could get more than one community at the end of this, of a long simulation, or using this persistence theory. I think we found relatively few cases where there was either no final endpoint community or there was more than one. So yeah, very few where there's two or zero endpoint communities. So that's instructive in a way that there really is usually only one final solution given a starting species pool. We wanted to know when the best time, how long you should sample for before you had a good idea about who was going to be there in that final endpoint community. So we looked at sampling over the first couple of years, and that really tells us very little useful information about what's going to be there at the end. First 5 years, we're getting closer to what's going to be there at the end, and maybe after 10 years, we're getting close to some acceptable answer, allowing for some uncertainty around that in terms of the correlation between the number of species, just the number of species, not the biodiversity Shannon index or something like that, of species that are there after 10 years. If we count them in the first 10 years, are they still going to be there after 100 or more years? That's the questions that question we were asking here. I haven't gone into how that varies according to the global species pool that you're working with, or the number of species that could be in the food web. So that's something we could dig into and colour these black spots a little bit differently for different sizes of species pools, and gain a little bit more information about what happens according to the community size that you're working with. I mentioned we sometimes got extra species in there that didn't appear in the final community. We also had to account for the fact that it took the finalists, the species who were going to win out and be present at the end, sometimes it took them longer to appear and be detectable in the community. So sometimes they appear to be missing from our sampling at the start in the first 2, 5, or 10 years as we move. And that's a fairly consistent answer as we move from smaller to larger communities on the left to the right there. So we might be missing species in the beginning that are going to be important over the long term. I think that's the way to summarize that. I'm going to skip over that because I'm concerned about the time. So quick summary is answering the questions we started with. How variable is community assembly after we restore a seagrass meadow or rewild a marine desert, or some other way of thinking about this? There's some patterns that are very consistent across small to large food webs that are built upon the foundation species of the seagrass. But the larger global species pools, the larger food webs, retained a lower proportion of the members in the final persistent set of species. We can play around with the variability across replicates and the uncertainty in the parameter distributions with these sorts of models, and just say, well, actually, if we incorporate that uncertainty in the sampling that we've got from field data that we're basing some of the parameters on, we can play around with that and find out if that's going to change some of the broad general conclusions that we might try and draw from this. So that's a nice way of incorporating mathematical modeling into some of these questions related to real world data. We can ask how quickly the communities recover following the introduction of the foundation species, and a rough rule of thumb is that we should be targeting 10 to 20 years after the initial reintroduction of the foundation species to have a reasonable idea of the final set of species that's going to be there. Maybe it's not worth spending money in the first 5 years when you're going out sampling. Wait until after 5 years, then go and see who's there. And I haven't answered the question about recovery patterns varying across trophic levels because I haven't looked at it yet. But it does seem to be a really interesting question that ties in with some of the other points that speakers have made today and in previous seminars on this workshop.